Hi class, today we're going to be looking at the mechanism of CF. So that's what exactly is happening within the cells of patients who have cystic fibrosis. So the first thing we want to do is just think about what we already know. So what background information have we gained already? So we looked at our lab, and I just want to talk about what we learned within our lab when we modeled cystic fibrosis. So we made two cells. So remember, we made our control cell or a normal cell, and then we had our cystic fibrosis cell. And if you remember, in our normal cell, we should have had the 0.9% salt solution inside the cell and our 0.9% salt solution outside of the cell. And this was modeling our normal cell in an isotonic environment, so having that nice equilibrium, okay? On the other hand, we should have made our cystic fibrosis cell, which was a hypotonic environment. And inside our cell, we should have had our 20% salt solution. And our external was still the 0.9% salt solution. Okay, so what we saw then was here we should have had water, okay, moving both into and out of our cell in what we would refer to, I'm going to zoom in actually. Okay, so water moving into and out of our cell, and this is a nice, equilibrium. So just to bring that word up again, equilibrium state. Oops. Okay. On the other hand, let's see if we can focus a little bit better. On the other hand, in our other cell, okay, we had water moving in at a much higher rate into our cell than it would be leaving. Because it's trying to get in there and space out all of those little salt particles that were in there. So this massive influx of water, and we should have seen an increase in the mass of our cell over the 20 minutes that we observed it. Okay. So then we also know, let's zoom back out again. We also know that cystic fibrosis patients have this thick and sticky mucus, but really want to know why. Like, what is it that's causing that to happen, right? So trying to make this connection between what we did in our lab and what's going on with these cystic fibrosis patients and that thick, sticky mucus that gets trapped in their bronchial tubes in their lungs is really hard to eject or really hard to cough up. So let's take a look again. So this should look familiar. Okay, so looking really familiar, hopefully. We'll start on our normal side, so we'll just cover this side up. We're going to look right at our normal cells. So we said that if we zoom in, we've got our, this is supposed to be my lung. This is supposed to be my bronchial tubes. Okay. So we're looking at the zoom in of that bronchial tube. Within that bronchial tube, we have a line of cells. So we have this tissue here made up of a bunch of different cells. We see that some of them are goblet cells and some of them are not. So it's not that every single cell along this tissue or inside the bronchial tube is a goblet cell, but some of them are. So we've got 1, 2 goblet cells right here. We know that the goblet cells actually eject or create this mucus. So they produce all this nice flowing mucus to make sure it traps dust or traps bacteria before it gets too far into your system and helps lubricate the airway as well. So that covers this cell in the cells in the bronchial tube. It covers that tissue nicely to keep it moist. And here we should see this nice balance. So the mucus should be fairly watery. Okay, so fairly watery. And we should see a nice balance of water, so moving into and out of our cells, just like in the model. So a nice balance of, okay, nice balanced equilibrium state. So our cells and the surrounding area, even our other cells, so even this cell, should have a nice balance with the, okay, within that environment. So there should be mucus just covering the cells, and there should be a nice balance of water in this isotonic environment. So this is an isotonic environment. Perfect. Now, on the other hand, we have our cystic fibrosis cells. So we're looking again, so same thing. So start off with the same thing. Here we've got a zoom in again to that lining of the bronchial tube. So here we go. I'll label it just in case. I know my drawings are not always the best, but here's my lung. They're supposed to be my bronchial tubes. Okay, perfect. So we're zooming in here. We see this tissue. We see there are some other cells, but then we have two goblet cells that we're pointing out. And here we are going to see something slightly different. So here's our mucus. Okay, we know that our mucus contains water. Okay, contains water. But remember, these cells, so the goblet cell, and we're just going to add with the 20% salt buildup. Okay, and with that 20% salt buildup, well, that's going to start sucking in the water within the mucus. Okay, so it sucks in that water into those cells. So that being the case, our mucus then is going to end up a little bit different. So zoom in again, okay? So zoom in again using some of the things that we looked at the intro to our lab. So this is supposed to be my goblet cell again, just impacted by CF. I'm seeing those chloride ions. So remember the chloride ions, we've just been calling this salt, and that's fine. So we've been referring to the chloride ions as salt. So we've got all of those chloride ions just building up, trapped within the cells, and this is causing that water to move into the cells, and this is giving us this hypotonic environment. So I'm not getting close to the edge of the paper, so I'm trying to hold it down. So this is creating this hypotonic environment, okay? And we're pulling in water. All right, so let's move on to our next one, okay? So we need a little bit more information. So here, I'm going to zoom out. I got a little crazy with my drawing this time. So I made a giant goblet cell, and it says, what is happening? So, looking at the cell that I created, it starts off, we see the very top, just like we saw, it has these nice little appendages. These are the, vesicles containing mucin or mucus for our purposes, or mucus. And remember, they're going to move toward the top of the cell, and then they're going to become expelled by the exocytosis to cover the cells. Okay, so they'll be expelled there by exocytosis. Okay, as we move down to the bottom, so this should look very familiar to our other drawing that we did of our goblet cell. We have some more structures. So here we have these two pancake-like structures. So these are both the Golgi bodies. Okay, so one there, one there. There's another little vesicle. Okay, so I don't need to label them all. We labeled one already. So here's another vesicle, here's another vesicle. This is the ER, or the endoplasmic reticulum. These little tiny dots then are ribosomes. So remember, they're used to make the proteins. And then here we have our nucleus. Here are some mitochondria, because every single cell needs energy, so that's where mitochondria come in. Okay, perfect. And inside we have all this squiggly junk. So this squiggly junk, well, that's supposed to be the DNA. So that's our nucleic acids there. Okay, so we're going to start at the bottom and make our way up. So starting with number one. Okay, number one, things we must know about the mechanisms of cystic fibrosis. Try to get this perfectly centered if I can here without messing with the camera too much. So one, if you have cystic fibrosis, your DNA contains 2 copies of a bad gene. Okay, so by bad, I mean that it makes a non-functional protein or a faulty protein that doesn't work in the body. So where do you get these from? You get them from your bio parents or your biological parents, so whoever your mom and dad are biologically, that's where they came from. A gene, just to remind you, if you're not familiar with it, the gene is a piece of DNA with instructions to make a protein. So we're really just focused on this bad gene. And unfortunately, people with cystic fibrosis get 2 copies of the bad gene. So one from their biological mother, one from their biological father. Then what happens after that? So we've got this bad gene. So 2. Okay? The gene that we're talking about is supposed to make these chloride ion channels. So that's a type of membrane-spanning protein or protein channel that helps remove salt or these chloride ions from the cell. So that's what the protein is supposed to do, and the gene is supposed to give good instructions in order to make this protein channel. Now, if it works properly, then when we have salt that builds up within the cell for just regular reasons, so the regular processes within the cell sometimes can produce these salts, then the cell has a way of getting rid of it. So they can actually pump it to the exterior so that it's eliminated. And this is supposed to be, I'll label it, this is my chloride ion channel. So it just looks like one of those protein channels like we saw in the POGL, okay? So it just looks like that, and it's just to remove this type of particle, so just those salt particles or those chloride particles. Now, okay, in the people who have cystic fibrosis, I might try and zoom out just 1 minute. Give me a second here. I want a little bit more to visualize here. Okay, perfect. So in our cystic fibrosis patients, the protein is flawed, so that means it has an error, it's not working, it's not making these good protein channels that people need. So what happens to them is, because they have some sort of flaw, they end up getting trapped in the Golgi body or the endoplasmic reticulum. So they get stuck either here or here. That being the case, they never ever make it to the membrane, so they never make it to the external cell membrane, and they're never added to the cell membrane, so they're never incorporated there. Okay, they just don't get there. So this is creating a big problem. Okay, so big problems now are underway, because now we have our cell membrane and there's no chloride ion channels. So our chloride ion is going to get built up in the interior of the cell. So here I'm just showing that inside the cell, so this is supposed to be the inside, we have this increase in salt. So it's getting really, really, really salty in there. And there's no way for the salt to leave, because the salt is charged, and charged particles cannot cross through that phospholipid bilayer directly. Just so you know, this is a simplified version of the cell membrane, because there still would be other protein channels there to transport other types of substances, but there's no specialized protein in order to transport the chloride ions. So we're just showing that they are the ones getting trapped within that cell. Now remember, when we have those charged particles, the charged particles just cannot make it through that cell membrane, so they're getting stuck in there. And now the problem's going to build, all right? So the problem's kind of building and building as we go here. So we get salt built up within our cell. So now the cystic fibrosis cells are not able to eliminate any of that excess salt or that extra salt created through normal cellular processes. So it can't establish equilibrium. Okay, it's not getting back to that nice balanced state that would make the cell, fully functional and content, if you will. So it's not getting back to that state. Okay, it's just not getting there. It's not able to. So the salt will just keep building up and building up and building up. So now, with the salt accumulating or building up on the interior of the cell, the salt within the cell is causing the water to move from the mucus or get sucked in from the mucus to the interior of the cell. So it's sucking up that water just like we saw here. So I'm just going to go back real quickly. So just like we drew right here. So that saltiness is causing this water to get sucked in from the mucus into the goblet cells. Okay, so it's sucking in that water. So this is where the problem comes in. So why is their mucus all sticky and gross, and why is it not normal and flowing and hydrated like it's supposed to be in a healthy person? Well, it's not that way because the water is being sucked into those goblet cells to try to balance out the salt concentrations. So now we have this very thick, viscous, sticky mucus. And this mucus becomes really problematic because people cannot cough it up. And it also, because it's so thick and sticky, it can trap the bacteria and trap materials within the lungs. Whereas in a normal person, if you get something stuck within that mucus layer, because it's so thin, you can cough it up very easily. Okay, but the people with CF cannot. So then it gets trapped in there and they have all sorts of problems with that mucus, and then all the materials that get trapped in the mucus as well can lead to further infections within the lungs. So when you're taking your notes, I would like to see some pictures. All right, it doesn't have to include color or anything. The colors are just kind of for me. I like to add the colors so that we can see everything. But remember, you can go back and rewatch anything and let me know if you have any questions. But we're really thinking about that mechanism. So what's happening now within the cell membrane that's causing these people with cystic fibrosis to have this thick, sticky mucus? What's causing these types of breathing problems? So we're really trying to put that together for ourselves, okay? So when you're done, again, I am going to ask you to write a summary. The summary should be about 4 to 5 sentences about what you have learned. So not one sentence, but 4 to 5 sentences. So trying to make sense of it, construct some meaning, and write a short paragraph summary, okay? So have a good day, and I will see you in class. Bye.